It is a pleasure for me today to introduce the department speaker, Mike Behrenfeld from Oregon State University. As some of you, many of you know, we're right now running the Ocean Color graduate class, and Mike Behrenfeld and his colleague Toby Westbury are teaching this class together with me. So we're very grateful for you guys to come down and helping us learn all about Ocean Color. Of course, also great that you're coming here today to give the department seminar. I think Mike Behrenfeld has made a lot of impact on multiple fronts in science. For one, as many of you are aware of, he is the lead on developing net primary productivity algorithms and in general using remote sensing to understand ocean ecology and biodiversity. However, I think another thing I would like to highlight before we start is also Mike Behrenfeld's push to challenge sort of existing paradigms. And it produced a lot of papers that are really challenging the way we think about how climate change affects marine communities that have gone sort of against sort of current dogma. And I think that's sort of a hallmark of great science. Anyway, thanks for coming, and I look forward to your talk.
Thank you all for coming to this talk. I'm not going to talk about remote sensing today. I'm going to touch upon some other things I've been thinking about during the last couple of years of COVID, because I have a lot of time on my hands and I want to talk to you. And my brain is spinning and I'm talking to myself a lot, and that leads to interesting places. So we're going to talk about some ideas I've been having on biodiversity, the plankton succession, and coexistence, or the mechanisms of coexistence. And this story begins with a project called NAMES, which stands for the North Atlantic Aerosol and Marine Ecosystem Study. And this was a project that was funded by NASA. It was a 5-year field study in the North Atlantic, subarctic North Atlantic. And we were going out to look at the influence of plankton on aerosols and clouds, and also to study what drives the phytoplankton bloom in the subarctic Atlantic. It's the biggest bloom of phytoplankton in the world. It's a huge bloom. And the study had 4 cruises. We went out in the wintertime, which is kind of nasty in the North Atlantic, and in the spring, during the beginning of the bloom. And then we targeted 1 cruise for the climax of the bloom, and then we had a fall cruise. And what I had expected when we were going to go do this bloom climax is to see this, to see a bloom full of these big diatoms. And we didn't. And this was the classic bloom, but we didn't see the big diatoms. And so that made me start thinking, well, what is it about phytoplankton ecology that I'm just not getting yet? And so this started leading me down a long rabbit hole of thoughts about biodiversity and plankton ecology. Led to 5 papers, which I refer to as my COVID collection. And at the end of the talk, I'll give you a list of those papers if you're interested. Maybe by the end of this, you won't be. So let's just think about diatoms for a second. Diatoms are one of the most recent additions to the geological record. We have fossils of them that are about 190 million years old, and other evidence suggesting that maybe they go back 240 million years. When the diatoms emerged, we already had armored dinoflagellates and armored haptophytes. So if we think about that, what is it that allowed diatoms to regain a foothold in marine ecosystems? Does anyone have any guesses? I mean, you're looking at a diatom here. Any thoughts of what might they have done that's different that gave them an edge up? Yeah.
Utilizing silica?
Yeah, so they have these gorgeous silica frustrables, and that might be important for what?
Protection from grazers.
Protection from grazers. That's one of the most common things that people say about why diatoms are successful. Anything else? You may not know this, but diatoms have really big internal vacuoles. So the big diatoms are more vacuole than they are cytoplasm. And so people have suggested that these vacuoles are important for luxury nutrient uptake. So they can suck up nutrients, and then when the nutrients get depleted in the surrounding water, they can use their internal reserves to keep dividing while other species are not. Okay? That's kind of the common explanation. I'd like to suggest that those are probably wrong. Okay, so first of all, why do we assume diatoms are successful? Well, if you actually look at it in terms of number abundance, they're really not that remarkable. Prochlorococcus is much, much more abundant by numbers. Diatoms are quite rare. In terms of global production, they're probably responsible for less than 20%. But where they really shine is diversity. There's more species diversity in the diatoms than all the other phytoplankton combined. Why might that be? So I'm going to throw a couple ideas at you here, and they are related to the frustule and the vacuole. And these two things playing with each other. So what I'd suggest is maybe the frustule's most important thing is that when you build a new diatom, you split the frustule, it's like a petri dish, you split it in half. And one of the daughter cells is the same size as the parent, the next one's a little bit smaller because they make the next frustule inside the bigger one. So after multiple generations, the size of the population keeps going down because they keep getting smaller and smaller. And so soon they have to go through sexual reproduction to reconstitute the original size. And all the other phytoplankton don't do this. So the diatoms have a much higher requirement for sexual reproduction, which I would propose is actually important for more rapid tempo of speciation. The other thing I would suggest is that this vacuole was really important in diatoms. The frustule gave it a rigid structure, which allowed big vacuoles to start developing because it could withstand the pressures of a big vacuole inside the cell. And that the vacuole was really important because it allowed, as species, as the diatoms started to diversify, they not only diversified in species within a size range, but they got bigger and bigger. Now, why might that be important? Why is getting bigger an advantage? That's Adam?
Less things to eat.
Yeah, so the bigger you are, you need bigger predators to eat you, which have more complicated life cycles. And also just handling times go up. So that's an advantage of the vacuole. And one of the problems of the diatoms is as the vacuole got bigger and the cells got bigger, well, they sank. Now, it's not good in the ocean, right? If you get to the bottom of the ocean, you're gone. So these big guys probably first started out in shallow waters, so they could be resuspended by physical processes. But then as evolution went on, the diatoms learned to use that vacuole to maintain their own buoyancy. So they used the vacuole and buoyancy control, and once they were able to do that, the big guys could go out and colonize the pelagic. The idea of nutrient uptake in the vacuole, nutrient uptake, is probably wrong. If we actually look at lab measurements, diatoms can't make any more cell divisions after nutrients run out than any other species. All the other groups do the same thing, just not with vacuolar storage, but it's organic storage. So we can handle that alone. What diatoms are famous for is blooms, big blooms. This is an example of a big bloom in the subarctic Atlantic in the background picture. And on the coast of Oregon in the spring and summer, the water's brown if you look at the lake. There's a lot of diatoms there. Right? But the ability of diatoms to bloom is not a feature of diatoms. It's a feature of specific species that have developed or adapted a blooming strategy for success. Okay? So since it's not a general feature of diatoms, we probably shouldn't try to attach blooming necessarily to the vacuole and frustule, but some internal physiology. What might that be? So if you look in the literature, when we talk about blooming diatoms, we often say, well, diatoms create blooms because they have high maximum division rates, right? Does that make sense? Well, the maximum division rate of a phytoplankton is determined by the dark reactions of photosynthesis, the Calvin cycle. It's the rate that they grow when they are light saturated and nutrient saturated. But if you actually look at blooms, they don't start at high light, they start at low light. So what good is a high maximum division rate if you're not growing at a maximum division rate? So what's the point of a high Emax? Let's try to answer that question. Now, if you think about growing in the mixed layer of the ocean, you can envision a cell circulating around in this very variable light environment. So what you're looking at photosynthesis on the x-axis, time of day, and different depths, okay, going down. Most of the time, when these cells are in this mixed layer, they're light limited. There's only a few moments during the middle of the day near the surface where they're light saturated. So you can ask the question, if I have some cell that has a relationship between photosynthesis and irradiance like this, if I want to grow faster, do I invest in more pigments to absorb more light in this light-limited environment? Or do I invest in more dark reactions, the Calvin cycle, the Max? Now let me guess. It's easy to calculate, so here's my little game. I'm going to do this exercise. I'm going to assume some realistic submarine light conditions for a mixed layer of 20 or 50, 100, 200 meters depth. I'm going to run a baseline calculation of growth rates for the simulated light environment based upon the black line that I showed right there. And then I'm going to say I have two different strategies. One, I'm going to increase my pigment by a factor of 2. That's the orange line going to the left. I absorb more light when it's low light. Or I'm going to increase my Kelvin cycle capacity by 2, the blue line. And then say which one is doing better compared to the black line. Everyone follow me? Yeah? Nods? Okay. It's a surprising result. So what you're looking at here, I allowed my incident sunlight to go from 1 to over 40 moles of photons per day. 1 is almost overnight, it's really, really low. Okay? The green, orange, and red lines, that's the enhancement over the baseline if you invest in the Kelvin cycle. And the blue line, the gray lines, those are if you invest in pigments. So for most incident surface light levels, you're better off investing in the dark reactions. It's kind of weird, isn't it? What they're doing is they're making hay when the sun shines. When they get near the surface, toward the middle of the day, they are cranking on photosynthesis, okay? So they're taking advantage of when the sun is shining. Now here's a really stupid question, which I really like to ask, because I can sometimes answer them. Why is it important to divide faster? What good is that? It seems like a dumb question. Well, if you're growing faster, you're going to bloom faster, right? Well, here's the reality of it. If you compare the rate of biomass accumulation, or the blooming rate on the y-axis, to the actual division rates of the cells, there's no relationship. You can be blooming, which is above the horizontal black line, at the same division rate that you're declining at another time of the year. So there doesn't seem to be a relationship between the division rate and how fast you grow. Isn't that weird? So why adopt some sort of strategy of having this high max if it doesn't matter? What we have to remember about the phytoplankton is the phytoplankton live in a system that is perpetually coupled between their division and things eating them. So it's a growth-loss balance. And it's the balance between these two rates that define how fast the bloom is accumulating. So our accumulation rate, RT, at some time, is equal to the division rate, μt, minus the loss rate, RLT. Now, if I tell you that the growth and the grazing is tightly coupled, but there's a slight time lag, we can rewrite this equation. We can say that the growth rate, μt, is going to be minus the loss rate that's equal to μt sometime earlier, or μt minus μti, where I is this time lag. Okay, is everyone following me? Okay. Which is interesting about this equation is that it's saying that the accumulation rate, RT, is proportional to the rate of change in a rate of growth. So you have a term that's dependent on data to the -1 on something that's data to the -2. So the answer to the question about the division rate thing is it's not the actual division rate that matters for blooming. It's the rate of change in the division rate. And the reason for that is because as long as the phytoplankton are growing faster and faster every day, their grazers can't catch them, right? They're a step behind. And so that difference between the growth and the loss allows them to bloom. What you're looking at in the graph here, I guess I lied, I am using some satellite data. The white circle line, this is multiple years of satellite data for a polar region, a north polar region, is the rate of biomass change. That's how fast the bloom is happening. And the red line is simply a calculated or expectation of the rate of change in the rate of division. And that's a perfect match in satellite world, right? That's really good. So what it's telling us is that bloom-forming diatoms, because they have a high mu max, can accelerate their division rates faster than other species, and that allows them to get out ahead. Does that make sense? Okay. And diatoms with high mu max can accelerate longer as well. So they can start accelerating, they keep going longer, because the other species are hitting their mu max earlier, so that the diatoms keep blooming and accumulating. What actually turns out to be the real important thing is the difference between the minimum division rate and the springtime maximum. That's kind of their acceleration range. And how big that is tells you something about how big the bloom is going to be. Okay? Okay. Oh my goodness. All right, so if we look at measured laboratory maximum division rates, the left axis, as a function of cell diameters, what we see is that the relationship is not a simple allometric relationship with size. It's complicated, I won't get into all the details, but essentially, all of the species fall below some envelope. It's not an allometric curve, it's an envelope, okay? And it's the bloom-forming species that kind of follow up toward the top of that envelope. So you might ask yourself, you know, if you look at the small end here, they have much higher maximum division rates than the larger ones. So why do small diatoms tend to bloom before the large diatoms? And how can large diatoms actually achieve a bloom with such a low max? Any thoughts? Yeah, there's, in my equation, the I is bigger for the bigger cells, the time lag, okay? So you can run a simple exercise. You assume a mixed layer with improving growth conditions in time, it's shallowing, getting more light. We have three different diatom sizes, small, mediums, and large. We assume that Emax, the large ones, are a factor 2 lower than the small ones, but they have a time lag that's a factor 2 longer. And what happens in that simple simulation is you get the small, medium, and large ones essentially reaching the same maximum biomass, but it's timed differently with the small ones first and the large ones later. Okay. So after thinking about this, I started just wondering about the general concept of species succession in plankton systems. And how many of you heard of Margot's mandala? Oh no, really? Come on guys, Margot's mandala? This is famous stuff. All right, maybe I'm older than I want to admit. So Margolov in the 1970s was proposing, a mandala, a conceptual diagram of succession. And he envisioned that the driving things for these successions were the nutrients on one axis and turbulence on the other. And his rationalization here was that large diatoms, because they sink really fast, need a lot of turbulence in order to remain suspended, and a lot of nutrients because they're big to grow and achieve biomass. And then as you go to less turbulence and lower nutrients, you get to smaller diatoms and caucasus and dinoflagellates. Since Margot's time, there's been some developments. We've learned that diatoms, no matter how big they are, can remain neutrally buoyant. So we don't need the big turbulence to keep them in suspension. We also know if you go to the subarctic Atlantic in the winter, I hope you never do, but if you do, you'll see that the population is not dominated by diatoms. It's dominated by little green guys, right? That's when you have the maximum nutrients, maximum turbulence. It's nothing like Margot's radiation. The diatom bloom actually happens when you have a lot of light, lower nutrients, and low turbulence, okay? And then we already talked about the size-dependent succession of diatoms. So I would propose a new mandala, and we're going to start this mandala with this stable solution. I'm suggesting that there's this kind of attractor, this stable attractor that phytoplankton populations go to as the environment becomes more and more stable. And so on my axis now I have the duration and the magnitude of change in living resources. Essentially the axis is saying how big is a perturbation in their growth environment. And then the y-axis is the growth condition getting better or worse. So we're going to perturb our stable system, and the first thing that's going to happen is these little guys that can really accelerate, they're going to dominate, right? But they're going to hit their mu max sooner, And if they hit their remax and there's still resources around, those guys are gonna get caught by their grazers and go back toward the stable. Large guys will show up until they deplete the nutrients. They go back, and over the course of this bloom, they can build up organics that other mixotrophs can use to allow those populations to develop. Of course, you can always short circuit electrical diagrams. One short circuit might be like in the Southern Ocean. The bloom starts going, but they hit iron limitation, and you never get to the bigger successional stages. You can also imagine that my thing stops working. Oh, there it goes. And maybe during the successions, larger ones, you run out of something special like silica, right? Now the large diatoms can't reach their maximum. They go back toward their stable solution. And this might open up a window for some other smaller things like coccolithophores, because you might have these grazers that were planning on eating big diatoms. They never come to fruition. So they become carnivores instead of herbivores, opening up a window of opportunity for things like coccolithophores. All right, how are we doing? Everyone still with me? Okay, so I want to get back to this idea of a stable solution. If we go out into the ocean and we measure in the oligotrophic, mesotrophic, really productive areas, the phytoplankton size distribution is a continuum everywhere you go, okay? And between a stable oligotrophic and a stable mesotrophic, the slope of that size distribution on a log scale is really not that different. You're essentially taking this curve and you're moving it up, okay? Pretty much. And then as you go to even more productive and perturbed system, you tilt that up, right, a little bit. And if you actually calculate the biomass associated with these three systems, you see you go from a biomass dominated by very small guys in the red line to the seasonal blooms, which are dominated by big guys. So I would say that under stable conditions, our attractor is for this very conserved size distribution with a slope of between -4 and -4.5. So across all nutrient regimes, a full size distribution of phytoplankton can be found within any given size bin. There are many coexisting species. So each one of these size bins, there are many species that are coexisting. And no matter what the nutrients, you always have the full size distribution. How can this be? And this gets us to, how many of you have heard of the paradox of the plankton? I didn't get many mandalas, but come on guys, paradox of the plankton, okay? Classic stuff. So this is, classic neo-Darwinian thought about biodiversity. It's first stated by Evelyn Hutchins, actually in the 1940s, but the classic paper is a 61 paper. And he says the problem that is presented by the phytoplankton is essentially, how is it possible for a number of species to coexist in a relatively isotropic or unstructured environment, all competing for the same sorts of materials? According to the principles of competitive exclusion, one should essentially outcompete all others, so you have one species left. Okay? Does that make sense? Prediction. After this, there were actually experiments done to demonstrate competitive exclusion, and a lot of these are done on plankton because it's an easy model system. This is a classic paper by David Tillman, where he looked at two species, Astronella and Cyclotella, and he grew them together in different ratios of silica and phosphate, right? And depending upon the ratio, you could get the Cyclotella to dominate, the first graph, the Asteriella to dominate, or if you get the mixture just right, this kind of Goldilocks kind of stuff, right? One of the bears or whatever. If you get it just right, I forget those stories. You can get both. But basically the conclusion is that the number of coexisting species is going to be equal to the number of unique limiting nutrients. Okay. That, of course, is in contrast to nature, right? I just told you that there's many species in many different size classes in the full size spectrum all the time. But our models often give us Tillman-like results. So this is a modern ecosystem model of the ocean. What you're looking at is on the color bar here, it's the largest size phytoplankton that remains in the model. So you can see there's these big blue areas, and the only thing you have are tiny, tiny phytoplankton. Everything else is extinct. So this looks very much like competitive exclusion. And they often look at these results and talk about this R-star rule, but because I'm short on time, I'll skip through that. Okay, so most of these studies looking at coexistence assume the principles of competitive exclusion. What if that's wrong? If that's wrong, that basic idea, basic neo-Darwinian idea is wrong, what do we do then? And why might it be wrong? The first piece of evidence suggesting it might be wrong is this paper by Edward Holbert. And what he did is something very simple. He took measurements from the Sargasso Sea to estuarine population phytoplankton, only looking at the macrophytoplankton, calculated the distance between the cells on average. And he concluded that the cells are so far apart, it's impossible for them to compete for nutrients, because the nutrient depletion zones around the cells don't overlap. And he said, well, this is the solution to the paradox of the plankton. So if we actually look at this numerically, if we go from very oligotrophic waters, less than 0.05 micrograms of chlorophyll, to very eutrophic, 50 micrograms of chlorophyll, the size distribution slopes of the phytoplankton range from -3.3 to -4.5, and individual cells are separated by over 100 to 400 body lengths from each other. That's a long ways apart, guys. It's like competing with somebody on the other side of a football field for your oxygen, right? That doesn't make any sense, okay? Phytoplankton only occupy 0.003% of the volume they're in, up to 0.002. So I'm trying to draw a picture in your head now of a world of phytoplankton that looks like that upper picture, but even more so. Cells are far apart and the diffusion boundary layers are far apart. So if we don't have direct competition for nutrients, how do we explain the structuring of ecosystems in pelagic? Have you ever heard of the neutral theory of biodiversity? Anyone? Got one? Yay! Yay, thank you. Okay. Neutral theory is a concept about structuring of systems in the absence of competition or selective pressures. It was first introduced in the evolution of genes. So it was a gene evolution theory first, started in the 1920s. And it didn't actually get adopted by anybody in ecology until the late 20th century by Caswell and Stephen Hubble. And Graham Bell stated in a paper, it was kind of a funny comment. He said it was really surprising it took us so long to grab onto this because, you know, we should have jumped on this earlier. He said perhaps ecologists find it difficult to accept that the differences they see so clearly amongst their species have no functional significance. Perhaps it's easier for geneticists dealing with spots on the gel to be inclined to neutralism. So the most impactful neutral theory publication is this book here, Hubble's Unified Theory of Neutral Theory of Biodiversity and Biogeography. It's been cited between 5,000 and 10,000 times, depending on what search engine you use. What's surprising is that of those citations, only about 2% are in the marine literature, or in the aquatic literature altogether. So if, you know, ecologists in general don't like to think about neutral theory, we were really bad about it, right? Up until 2005, we were only citing one paper cited this book per year. We're up to 15, huh? Oh, good job, guys. All right. Okay, a little briefing here on what neutral theory is about. So Hubble built his theory around ocean or island biogeography from McCarthy and Wilson. And he has three fundamental assumptions. The first assumption is that all individuals in a community are equal competitors. They have exactly the same probabilities of giving birth, dying, and migrating. Second assumption is that when an individual dies, it is randomly replaced by any species in the community with a probability proportional to the abundance of that species. And the third, which is called the zero-sum constraint, is that landscapes are saturated with individuals. So if one thing dies, another one will replace it, but you can't get more. Okay? And the predictions of the theory are that without immigration, over time, you will end up with one species. But with immigration, you can have a stable coexistence of species. It's essentially saying just random processes can give rise to structure in ecosystems without any competition. Let me get just a visual of what this might look like. So what I have here to start is I have 4 ecological communities, and the meta community is the sum of all 4. And now with every generation, I'm going to kill 50%, I'm really a mean person, I'm going to kill 50% of them. And then they're going to be replaced randomly with the ones that are still in the boxes in proportion to how many of those are randomly left. So in 10 generations, you can see that our community is getting less diverse. The different colors represent the 4 different species. And in 100 generations, we're down to monocultures. And it's random which one actually wins. In 2 cases here with the gray, 1 blue, 1 orange, and none of the blacks survive. If we do the same thing, but we add just a little bit of immigration between each one of the boxes, a very small percentage, what we see is that we do lose some species, but over individuals, but even over the course of 100 generations, we maintain our diversity. The reason for this is that these statistical random processes are happening independently between each one of my 4 ecological communities. And so you can't predict who's going to dominate in each of those boxes. So if you mix it with the migration, you can always seed the places that one species got lost again, and they have an opportunity to become dominant over time. And it's the independence of this process in space that's important. So let's get back to the plankton. Okay, here's my game. I'm going to start out with 3 populations, 10,000 individuals, 100,000 individuals, and 1 million. I'm going to start out with 10,000 in unique species, all of equal fitness. I'm going to kill 50% and do replacement in proportion to the remaining abundances. Can you predict what's going to happen over time? I have no immigration. What's that?
Monoculture.
Yes, that's exactly what happens. Over time, you end up getting on this slippery slope of extinction, and it's the same slope irrespective of what the population size is, and eventually they all go to 1. You're the one who read the book. Okay? Oh, way too fast. I must have kept my finger on the, okay, so that was the solution there. Now, if we simply add a very slow rate of immigration, we end up with, instead of a monoculture, steady state biodiversity that's directly proportional to the size of the population. The problem here is that if you look at when these 3 curves first bend over, the first stochastic loss of a species is directly proportional to the population size. So if you apply this idea to the phytoplankton, where my biggest population of 1 million individuals can easily be found in a couple mils of seawater, and you apply that to the body of water that's easily mixed within a single day, the time it takes for the first exclusion of a species is longer than life on Earth. So we have some pretty hard conclusions. Neutral theory places no constraint on the plankton diversity. And because it can't get rid of species, immigration isn't important either. And so Hubble's prediction of the diversity equal to the population times the immigration rate, that last equation, is not true. So now we're kind of, I shouldn't say screw, should I? But I just did. Never mind. We're in a tight spot here, guys. We have no competitive exclusion because they're too far apart, and neutral theory doesn't help. So what do we do? So as I mentioned earlier, it's the independent stochastic evolution of these populations that's critical for neutral theory, and it's why it doesn't work in plankton. We need something that is causing a shift in populations everywhere at the same time in the same way. So think about this, just for a minute. Think about some plankton community, maybe the picoplankton, some size range, that's being grazed on by some specific grazers. The cells are far apart. However, within that population, we have some species that have 1 division rate, some species that are a little bit better. And when that population keeps getting turned over by the predators, the ones that are a little bit better keep having an advantage each generation. So here's the new game. 10,000 individuals, initialized with 200 species. Each species is assigned a division rate, a fixed division rate, between 0.6 and 1 doubling a day. And then the rest is the same, kill 50%, replace blah, blah, blah. And this is what we find when we run that simulation. So this is just some of the species. I don't have all the lines, but each line represents one of my modeled species. And you can see the blue line pops up and then it goes down another color. That's ecological drift. That's just the stochastic process going on. But as you go along to the right of this graph, if you actually count the species, we see that the richness of the populations drops quickly, and then it starts flattening out and becoming pretty stable. And we're looking at 3,500 generations there. What's happening is this, what I call trophic exclusion, that you have this relationship between predators and prey that is always pushing to narrow the fitness difference between your species. So you're always pushing toward the most fit for this community. It's pushing these lines from the beginning, which is the gray, 20 generations out to 1,000 generations, which is the green. And so what the population is doing essentially is it starts out with a very strong trophic exclusion process where predator-prey interactions selecting for the most fit starts wiping out species. And as you go further to the right, the population starts becoming stochastic, ecological drift. Okay? All right, what are the points that I want to make? Okay, so one of the things that's important here, this is a very simple model, but because as you get more and more equal fitness, it becomes really hard to get rid of more species. It means that the fitness of the different species is integrated over time. It exceeds the annual cycle. So in order to remain in the community, you only have to have an average fitness over the entire multiple years to remain in that community. And what I call this is the neutral niche. It's trophic exclusion and a neutral niche. Because each one of these populations that's being selected for is being selected for its specific size classes. Because grazers on small phytoplankton are small grazers. And different grazers are eating different phytoplankton populations. So each one of those grazing classes, which is the zooplankton on the right gray, is feeding on its own size distribution on the left. Each one of those specific predator-prey pairs is its own biological niche. And within those niche, processes are largely neutral. Okay? And it's the proportionality, rather than the absolute size range of the grazing, that causes the size distribution I talked about earlier to be a -4-ish slope rather than a -3. If they were absolute size grazing ranges, it would be -3, but the 4 comes from the proportionality. How are you guys doing? Hanging in there? Okay? Yeah? Get some people nodding off. It's all right. It's afternoon. I just had an espresso, so I'm doing great. All right. So is this trophic exclusion thing significantly different than classical resource-based competitive exclusion? As I mentioned before, the classical theory would say that the number of limiting resources defines the minimum number of species. And in this trophic exclusion, the number of minimum resources is irrelevant. It's based upon the net growth rate or the net fitness of the different species. Under regular competitive exclusion, low resource concentrations, the most fit of all the species will be the one that's left. But in the trophic exclusion, it's going to be the most fit within each of the biological neutral niches. Is classical exclusion compatible with the spacing of cells of plankton? No. The trophic exclusion has no problem with that spacing. And another interesting thing is that we often think of size as a master variable in defining competitions. And that would be the case in classical competition, but in trophic exclusion it's not, because the size term is neutralized by these different size niches. So a little bit about neutral niches is kind of an interesting idea. When Stephen Hubble was developing his neutral theory, at the same time, Peter Chesson was developing another theory. And in Chesson's visualization of ecosystems, he could see how environmental factors would select for fitnesses. He called these equalizing mechanisms. But then he countered those with what he called stabilizing mechanisms. So the environment is selecting for a narrow range of fitnesses, but the ones that are kind of on the left side of that fitness range may also have some stabilizing things that help them out. For example, sometimes one species getting really dominant actually hurts that species more than it does another. That's a stabilizing mechanism. Now, other mechanisms that are at play is not just division rate. There's predator avoidance, there's chemical defenses, there's lots of different things they can do to help neutralize any fitness differences they have. So we can think of our neutral niches for the plankton as being essentially a reflection of, processes trying to narrow that fitness difference way down, and then other things that help compensate to keep your richness there. Seasonal variability and where you line up on the fitness scale is also one of them. Here's an interesting question. How neutral is a neutral niche? How equally fit are these species in a niche? Who says they are almost identical? Who says they're quite different? Okay, we've got more quite difference. We don't know the answer, but I want to throw a tantalizing, thank you Adam for this, a tantalizing piece of evidence that might suggest it's the former. So this is work from Adam's group looking at nutrient stress genes in Prochlorococcus. Many of you might know about this paper. This is for the nitrogen stress genes in Prochlorococcus. And what you find is that in environments where the nitrogen stress is relatively low, the gene cassette, or the toolbox they have, basically they're only using ammonia in urea. But as nitrogen becomes more and more depleted, you find species with nitrate and nitrite utilization genes, which is cool, right? But that's not what was cool to me. What was cool to me is that when there's enough ammonia, you got rid of the species that were carrying the nitrite and nitrate. Think about that. A couple of genes to do nitrate and nitrite use is enough to select against them. So I think it's a razor's edge. I think that this fitness selection by trophic exclusion is really sharp. Okay. So I hope that was fun. I'm going to throw out some parting thoughts. One slide per thought. Getting back to a couple of the things that I brought up and never fully answered. May not have fully answered anything, actually. We'll see. So part one, names. Why didn't we see the big diatom plume? So the thought that I had there was that if you look at the difference in the mixed layer depth between winter and the climax of the blooms, in the named study site, the winter mixing depths are actually shallower than they are toward the east. And what that would imply is that the wintertime division rate is higher in the west than the east. So the difference from the winter minimum to the spring maximum is smaller. So the small guys, which we did see, bloomed, but we didn't have a big enough difference over the year for the big guys to bloom. That one to come first. These are not facts, these are just ideas. Number two, I talked about these models that are giving us mass extinctions of phytoplankton in the low nutrient areas. Why is that? Is it this R star rule? The little guys are getting all the nutrients. Well, that's not it, because they're too far apart. Well, this map here that I'm showing is based upon this blue line model, where as you increase the S infinity, which is the nutrients in the bulk media, you progressively increase your diversity. But what I found is that the reason that the model's doing this is not competition. It's because the models have a loss term in them that's just a daily mortality rate that's not associated with grazing. They just die. Phytoplankton don't just die every day, right, for no apparent reason. If you take that one term out, you maintain all of the species diversity and size under all nutrient concentrations. You can even say, I don't want to get rid of that. I'm just going to say the probability of dying is some fraction for every generation. Keep them all again. So I don't think our models are all inconsistent with what we're talking about here today. We just need to tweak them up a little bit. We can get our diversity in there. One of the things Chesson talked about in his theory on diversity was that the question, the challenge in ecology is not to explain unstable diversity, but stable diversity. And what he means by this is that if you have some inferior competitor moving from this environment over to this environment, the fact that you see it there is not very interesting. It's going to go extinct sooner or later. That's unstable coexistence. The question is the ones that can remain together forever. Our current models are not set up to investigate stable coexistence because each one of our assigned phytoplankton is specifically assigned physiologies that make them different, right? But what we can do with those models, we can evaluate what is the relative importance of unstable coexistence, because we can use our ocean currents to mix different phytoplankton that are fit for different areas and see how much diversity we get enhanced just by physics. And that's what this picture is showing. So you can see where there's strong currents intermingling. We're bringing in different populations from different areas that are better fit for different areas, and you get enhanced biodiversity in these yellow and red areas because of just physical mixing of populations. So we could use this kind of model and compare it to observations in the field, and we could say, well, this fraction of the biodiversity that I'm seeing may simply be unstable coexistence, and what's left is the stable part. Here's a fun one. Hubble's book is called The Unified Neutral Theory of Biodiversity and Biogeography, and in fact, it does not explain either of those things. The reason why is because Hubble starts with the assumption that all of the individuals are equally fit. He doesn't explain how they became equally fit, and they become equally fit by competitive processes like trophic exclusion. So I showed you this curve of the division rates. Do you remember this? Division rates is a function of size. If you want to try to understand that better, this is the same plot except for it's got a log x-axis. What I've done here is I've calculated the division rate that is supported by simple diffusion to the cell as a function of cell size. Each of the gray curves going from the bottom to the top is a different nutrient concentration going from 1 nanomolar to 20 micromolar. And what this is telling us, first of all, is that even at 1 nanomolar, these little guys like Prochlorococcus are right back there, they can do their maximum division rate. And as you get bigger and bigger, you have to have higher and higher concentrations to get to that maximum division rate. And so we have now the orange dots are all those phytoplankton cultures that I showed you, plotted on top of those diffusion rates. And you can kind of envision how maybe the reason we have those different cultures lining up all over that space is because some of those species are evolved to conditions that never get high enough in nutrients to hit their maximums. But it's a very interesting question. How do these species that seem to be inferior competitors still exist in the world? And the last slide, I'm doing okay on time, is this interesting question. So I got done with all this stuff. Like, okay, so we have these neutral niches, we're selecting for them. And I said that the neutral theory didn't put any constraints on our biodiversity. Well, what constraint is there on the neutral niche? Is biodiversity in a given ecological neutral niche still unlimited? I don't have a solid answer for that, but I'll throw out a couple ideas. One of them is this idea that as we go along in time, a species might invent something new that makes them a little bit better competitor. And that's going to cause a large loss of species, of previously coexisting species. And over that process of loss, you're going to have additional inventions in other species to kind of come back up to snuff with the new invention. And what that does is it gives you these oscillations. And here's going out 20,000 generations. I'm introducing every once in a while a new species that's better, and then also in time, new species that have new adaptations, but they're equally fit. And so we can kind of help dampen any long-term trends with this up and down, knocking them out and going to some new ones. And then there's this idea of kind of time's arrow. Anyone read the book about time's arrow? This is kind of abstract, anyhow. The idea is that, if you think about it, we have these lineages that start way back here, and they, over long periods of time, geologic time, they're evolving these strategies to remain in the ecosystem. As time goes on, they become obsolete, but they can't go back to scratch one and start all over. They have to keep patching the stuff that they had before, right? So they're always adding a little bit to a framework that can't change anymore. And maybe that's another thing that helps constrain diversity in these niches, so they don't have unlimited avenues to go. The proportional genome size thing, I'll do this quickly, but it's kind of curious. So in eukaryotes, the number of coding genes is independent of the size largely. What determines the size of the cell is essentially the amount of non-coding DNA. And that non-coding DNA evolves in a proportional manner. So if this evolution of the non-coding DNA is driving size, and it tends to make cells smaller more than it makes cells larger, then the evolution of the coding genes on top of this non-coding evolution would push the rate of speciation faster to the smaller cells and slower to the larger cells. And if you compare, this is what the bottom graph is showing, these green and the green dots are the predictions of the evolution of size based upon the evolution rate of non-coding DNA. The orange and the blue symbols are the size distributions of species diversity measured during the ocean tar program. It's a perfect fit. Kind of intriguing. And I just want to say thank you to the people who, I'm very, very, very lucky with the people I work with. Lee Cartboss, if you might know, Emmanuel and I have been working together forever. Kelsey Bisson's a research associate in my group, Robert Malley's in my group, Chris Munt. Okay? Chris Munt is a brilliant geneticist. He works on wheat, but he's been working with Kim Halsey's been working with me for a lot of years, Peter Gowdy, Alan Milligan, and Graham Pierce. So I'm just super lucky in my colleagues. Thank you all for your attention. I hope that was interesting. If you're interested in the COVID collection, this is the list.
All right, we have time for a few questions.
Yeah, just a point of clarification, because I think I got a bit lost with my sauce there.
Sorry about that.
So are you arguing that diversity is due to trophic fitness? But trophic fitness is defined by the properties of the environment. So what nutrients are there, the temperature, physical processes. So therefore, nutrient concentrations matter not because of direct competition, but because they are a property of that niche, and therefore what the organisms evolve and adapt to use. So then you can have this competition, like say the Rhodococcus thing, because that needs change in genes, which makes them not fit for that niche. Is that?
That would have been a shorter version of my seminar, yeah. Yeah, that's pretty good. So if we look at the biogeography of plankton, there's very clear environmental sorting, right? You go to the Arctic, it's a different community, the gyres are different communities, so that's very much there. And as you go down and down and down into more and more, specific kind of environments, the temperature, the light, the nutrients, all that stuff, then you start focusing in on trophic exclusion, selecting for some group of phytoplankton within a size domain that have all come up with some solution to that environment that makes them equally fit, or some stabilizing thing helps them out.
So that would explain the latitudinal gradient.
Explain the latitudinal gradient as well.
That's interesting because we did a bacterial biogeography paper for the UAM. We actually saw higher diversity in the gyre. Goes against like much of the ecological theory. So that would explain that.
Yeah, if you have a really stable environment and a lot of time, you would end up getting more diversity in that stable environment.
Yeah, because they're partition.
And there's another thing that can contribute, especially in the bacteria, is metabolic networking can do a lot of really increased diversity. You know what that is? So that's where, for bacteria, they will lose core metabolic genes because they rely on those products being produced by other ones. It's the black queen hypothesis. They're basically taking advantage, and you start building these metabolic networks. So this guy is dependent on this guy, who's dependent on this guy, who's dependent on this guy. So you get 4 species instead of 1, right? So you get a lot of in the bacteria. It's a little more difficult for me anyhow to justify metabolic networks in the phytoplankton. because basically their primary source is plankton. They all do their own chlorophyll.
So
But there are some trade-offs.
heterotrophs might do that.
Heterotrophs do it really, yeah, they do it a lot, yeah.
So one of my beefs with that paradox is that it sort of assumes that they are using the same resources, right? Phytoplankton. And in reality, right, they are using a whole slew of similarly issued resources, right? We were mentioning the nitrogen, right? They use nitrate, ammonia, urea, and cyanide, and you can go on and on for a long time for the various forms of nitrogen that they could access. The same thing for phosphorus and iron balance, therefore, et cetera, right? So what we sort of maybe as a very high bird's eye view think of as a uniform resource environment for an individual is in reality a really, really diverse resource environment. That could also explain coexistence. That they're accessing those in different proportions.
I would argue that, in fact, it is totally compatible with this framework, because different species that are utilizing different types of resources in different ways to achieve the same overall fitness allows for enhanced biodiversity. But that fitness is being trained essentially by the predator-prey relationship. Right? So you can get to the same solution in many different ways, but you have to get to the same solution as a net. Integrated over the time scale of trophic exclusion, which can be a long period of time as you get more and more equal fitness. A long period of time. Thousands of generations.
Just a random question. I mean, how important do you think it is for evolution, like, you know, thriving in the euphotic zone versus like surviving deeper? And it seems like the benefits that give you a rapid growth in the euphotic zone might be ones that would kill you if you're trying to survive when it's dark and you get subducted deep into the ocean interior. I'm just curious.
So you're thinking about deep in the ocean interior, you're still talking about in the photic layer.
Well, no, I guess maybe this is really naive, but just that sometimes the organisms are, you know, traveling down and then they'll pop back up. Like a lot of the models assume any water packet that comes to the surface has the organisms in it, and then they can compete for the resources, right? That but I'm just curious if the survival is set by processing the euphotic zone versus deeper.
It's a really interesting question, Jim. So if we partition it first into the photic zone, we have the mixed layer and we have more stratified water below. The guys below are going to be dividing slower, right? But their loss rates are going to be slower too. So the enhanced division rate in the surface is coupled by enhanced turnover, which means the selective pressure for narrowing that fitness gap is really high. Down below, they have more time. If you take these phytoplankton and you take them below the photic zone, well, their division rate goes to zero by definition. And as long as there are loss processes, you're going to lose those most likely before that water comes up in a permanently stratified system. In higher trophic or higher latitude areas where the mixed layer depth goes from shallow to very deep in the wintertime, one strategy for a phytoplankton might be to create cysts. And those cysts can persist, they're not going to be eaten. They can persist for the entire winter, come back up again, and maybe start bloom. Now, what I argued in the diatom paper is that if you actually look at how much data we have on cysts that are being maintained down below, and a couple cysts come up, well, what are they relative to the fraction that are still active in the photic zone that are, right? So you could argue that's probably not that important. But if you're in a neritic zone where you have a huge amount of cysts sitting in the sediments and they get resuspended in the spring, that's a big deal. Because now you're starting out with a seed population that's much bigger. And so your bloom starts with a big one and goes even bigger, so you get bigger blooms.
Well, I'm going to cut it short a little here, but we have a reception starting now downstairs, and you'll have opportunities to ask Mike all the questions that you want, and also get a chance to talk to our prospective students that are visiting today, the prospective grad students. So I hope you'll all join us at the reception downstairs, and then we can continue the discussion. Thank you, Mike.